Okay, we're going to continue respiration with the steps of Krebs cycle. Now, the first thing that occurs in Krebs cycle is that 2-carbon acetate enters your Krebs cycle, joins with 4-carbon oxaloacetate to form 6-carbon citrate. Now, to diagram this, here's our acetyl-CoA that comes in from glycolysis. Coenzyme A is cut off. It returns to the cytoplasm. That leaves 2-carbon acetate that enters Krebs cycle, joins with 4-carbon oxaloacetate that's already there in the Krebs cycle to produce our first compound, which is 6-carbon citrate. So 4 carbons plus 2 carbons yields 6-carbon 6-citrate. Now, Krebs cycle is also called the citrate cycle because the very first compound that's formed in this cycle is citrate. Now, all of these chemical reactions that occur have an enzyme that allows that reaction to happen. In more advanced classes, you will have to learn the names of all of the enzymes that control these reactions. But for our purposes, we're just going to see what the end results of these reactions are, and we're not going to learn the names of the enzymes that cause them to occur. All right, the next thing that happens in Krebs cycle is that we've got a loss of carbon dioxide from citrate. This forms a 5-carbon compound called alpha-ketoglutarate. Now, you'll remember when a compound loses carbon dioxide, that's called decarboxylation. And that's a word you need to remember for the test. Now, the second thing that happens is that citrate is oxidized. It loses 2 hydrogens. It loses it to NAD to form NADH plus H, a bus for ETC. Now, citrate loses the electrons, NAD gains the electrons, it's reduced, citrate is oxidized, so this is a redox reaction. So to diagram this, here's our 6-carbon citrate. Here's decarboxylation, the loss of carbon dioxide. Here's the loss of 2 hydrogens. This is the oxidation gained by NAD, that's reduction, to produce a bus NADH plus H. And so what we're left with is a 5-carbon compound that's called alpha-ketoglutarate. Now, here's our 5-carbon alpha-ketoglutarate. To get from alpha-ketoglutarate to oxaloacetate, all of these reactions have to occur in between. There's a loss of carbon dioxide, decarboxylation. Now that cut us down from 5 carbons to 4 carbons, but we still have to make all of these buses and an ATP. So our compound, the ketoglutarate, is oxidized. It loses 2 hydrogens. They're picked up by NAD, making another bus for ETC, NADH plus H. Now enough atoms and electrons have been chopped off of these compounds that a lot of excess heat is produced, and 1 molecule of ATP is made in each turn of the Krebs cycle. That's called substrate phosphorylation. Then 2 more hydrogen electrons and 2 protons are oxidized off, picked up by a new bus called FAD, or flavine adenine dinucleotide, to make FADH2. That's another bus for ETC. And then lastly, 2 more hydrogens are oxidized off of our compound, picked up by NAD to make another NADH plus H bus. And that brings us down to 4-carbon oxaloacetate, which is present there to keep running the cycle. Now, in the end results, one term. First time an acetyl-CoA comes in, donates the acetate, you get these results. 2 carbon dioxides, here and here. 1 ATP, here. 3 NADH plus H buses. 1, 2, 3. And 1 FADH2 bus, right there. But now you remember at the end of glycolysis, we don't just have 1 pyruvate left, we have 2 pyruvates. So there are 2 acetyl-CoAs coming to Krebs cycle. So for the test, you need to know the total end results for Krebs cycle. So you need to know the total. That's 4 carbon dioxides, 2 ATPs, 6 NADH plus H buses, and 2 FADH2 buses. Now here's an interesting fact. You see this carbon dioxide that's produced here in Krebs cycle? The carbon dioxide that we breathe out of our body is mostly made as a result of decarboxylation in Krebs cycle, and of course also in pre-Krebs. So all of this carbon dioxide that we breathe out is a result of cellular respiration that occurs in the mitochondrion.